Let's start the presentation session of polymer material application for biomedical field. So let's start with group 1, Andy and Ezra. Please share your screen and turn on your video. The other group, please pay attention, and later I will ask you to ask question also to the presenting group.
So good morning, Pak Dedi, Tanita Grace, and Angeli, and my beloved partner Ezra. Um, so today we're going to talk about the synthesis. We're going to present about synthesis and characterization of gelatin PVP polymer composite scaffold for potential application in bone tissue engineering. We take the journal in this paper made by Rutusmita Mishra et al. And for the introduction, the traumatic skeletal dysfunction and fractures caused by injury or aging, and the repair and replacement of damaged tissue can be solved by bone tissue implant. Synthetic or natural polymers can be used due to its flexibility in their structure and easier modification for enhanced biodegradation. For the gelatin, it is a polymer derived from hydrolysis of collagen. It is biocompatible, biodegradable, and it mimics mimics the biological properties of bone matrix protein, such as collagen. For example, collagen. It is efficient absorb capacity. It is non-toxicity, non-immunogenicity, and yeah, it can be used for bone tissue and bone tissue engineering application. As you can see, this is the chemical structure of gelatin. So for the PVP or polyvinyl pyrrolidone, it is long known as amorphous nature. It is ready solubility in organic solvents. It is it has capability to interact with hydrophilic materials. It is thin. It is film forming ability. It has film forming ability and have adhesiveness. It has stability at various pH, resistivity towards high temperature, cross-linking, and good complex forming capacity and mechanical strength. As you can see, this is the structure of PVP. So, uh, they can be combined due to the it, because of it has a compatible and non-toxicity um, character, uh, and due to its poor forming ability, they uh, can be used for and bone tissue engineering. So how to make so the journal gives us the steps how to make the scaffolds. So for the PVC solution, it was made in the distilled water with constant mixing on a magnetic stirrer. And then the gelatin solution was made in the distilled water and then mixing it to combine a homogeneous solution. And then the gelatin PVP made to 1-to-1 ratio. Solution was mixed and stir it. And then the sodium bicarbonate was added to the homogeneous solution. For initiation, it is for initiation and foaming. The acetic acid was added, and then the acetic acid was added to it is to do to initiate a chemical reaction, and then the produced foam of the gelatin PVP was then casted in the pre-chilled polystyrene plate, and then frozen it, and then store it overnight. The scaffold was cross-linked with the glutaraldehyde to mechanical strength, to enhance the mechanical strength, and then the glutaraldehyde solution and the remaining acetic acid and the scaffold was washed by deionized water. The scaffold then frozen overnight, followed by lyophilization under a vacuum pressure, and lastly, the lyophilized scaffolds was cut into different size according to the experimental needed.
So these are the results of the scaffolds that they made. Next, due to the time, I will explain it quickly. So the promising properties that they have is they have highly porous that supports for proliferation and migration, and then they have good blending to make an ideal scaffolds with uh, gelatin and also the PVP. And then it is biocompatible, and it also have water absorption ability. Next. So in the ratio of 1:1 ratio, the diameters for the scaffolds to be found in the range of 43.05 until 362.5 micrometer. And for the minimum requirement for the bone formation itself is approximately 100 micro meter due to the requirement of this cell migration and transport for nutrients. So if it's too small, the cell cannot migrate, and if it's so they need big pore. Next. So for water, they have one of the ability is they can absorb water. The gelatin PVP scaffolds can absorb water and create great potential of hydrophilicity. And the swelling ratio percentage affect the pH of the medium from lower to higher. And this pH also affect the quantity of the nutrient transport for the cells. For example, is iron. If the pH is high, so they can transport more iron and other stuff. Next. So they also have mechanical properties. For example, is tensile stress strain that can be seen in the graph. So if the stress is low, they can produce also low strain and other and vice versa. And they also have compressive strength with which is 2.08 plus minus 0.02 MPa megapascal. And then after that, they also have zeta potential. What is zeta potential? So it used to analyze to predict the interaction of biomaterials to osteoblast. And the negative zeta potential of the scaffolds will be highly supportive for the adhesion and growth of osteoblasts during the osteogenetic differentiation. So because the previous because of the water absorption, they create the high pH environment that will affect the zeta potential to be negative zeta potential, and that also support can make the adhesion and growth of osteoblast. And then next, so the stability. comparing to the pure PVP or pure gelatin, the gelatin PVP scaffolds have high stability compared to the pure one. So you can see in the table that the gelatin PVP have higher degradation temperature, so it is can be called that it is stable. Next. And then we talk about the biocompatibility. So to test it, they use C3H Tanti 1 per 2 cells. It is the cells to test the biocompatibility, and it is found that during 3 days, I think, it already covered the surface of the scaffolds that indicate that it has cytocompatibility property. And next, to test the biocompatibility, they also use a chem assay of a fertilized egg. And as we can see in here, so they put it what's it called, the scaffolds to the fertilized egg, and the scaffolds did not elicit any reaction in the chem, and the blood vessels were intact within like 3 days, I think. And then next, the last one is for further development. The author said that it should be tested in vivo animal models for better integrity with host tissue system. So they did not test it in animal yet, but later they said that they will do that. And that's all from us. Thank you.
Thank you, Ezra and Andy. Please ask question. Anjali and Tanita.
Yang tadi yang CH3T, what cell is that?
It is similar to the MSC cells. So they said that it can be tested to analyze the biocompatibility of the product that they want to test.
Okay, thank you.
Any other question?
Welcome.
Maybe Anjali.
For the polymer, can the PVP be substituted for other polymers? Maybe.
Andy, mau jawab?
I think it is possible with the similar character can replace PVP, but I don't
Yes.
remember the
Yeah,
characteristic of
we think the same. So it maybe it can be replaced, but it also have to have similar properties or even more better properties than PVP.
Okay, I have one question. So actually, what is the function of the PVP here? To have a binding between the ceramics particle in the implant, or what?
for the porosity.
Increasing or decreasing porosity?
Increasing, sir. So actually PVP is important in component in this scaffold because if it's just it increase the properties like porosity and also the stability and
Yeah.
I think it's that it has benefits like that. So as I mentioned before about the stability, if it's just pure PVP or pure gelatin it doesn't produce enough stability for the scaffolds. But if they combine to each other, they create better properties like that. And also they are cross-linked to each other, so yeah, they are strong.
Okay, so another question is that basically they are doing in vitro only characterization for the biocompatibility. They do not use yet in animals, so they use cell culture to test their biocompatibility. Is it so?
Yeah, they use yes, a chem of fertilized egg, but they are planning to test it on animal too one day, but I don't know when.
Final question is, can we make similar thing in our lab?
I think it's possible. Actually, I think it's possible because if there are equipments, because
and money.
and money.
I mean, do they need?
Because
does it need a special equipment that we haven't got in our lab?
pressure equipment, I think, because it has to be wait, I forgot. So it has to be in under vacuum pressure, vacuum condition. Yeah. So I don't think that our lab has that kind of tools. But yeah.
Well, depends on the pressure. If it is low vacuum, maybe we can.
Oh.
There is already equipment. If it needs high vacuum, then we are not capable of yet. Anyway, thank you, group 1. Let's move further to group 2 because time. Thank you, Andi and Ezra.
Thank you, sir.
Thank you, sir.
So, yes, Tanita and Angeli, please turn on your video. And
udah kelihatan?
Ya.
Oke, wait. Angeli.
Okay, so good morning, everyone. Good morning, sir and friends. Today, Tanita and I will present about FIMA and boric acid composite hydrogel as soft contact lens material. Next. Soft contact lenses are a type of prosthesis used by millions of people all over the world to correct visual disorders, changing eye color, and to treat corneal diseases. This is the reference we use, which is from the Journal of Applied Polymer Science. Next. So the first thing I will discuss is the materials and fabrication. The fabrication method is called the polymerization. So first, the cross-linking agent, which is boric acid, Ba, is dispersed in 10 milliliter of 2-hydroxyethyl methacrylate or HEMA solution with different percentages of Ba. The variations can be seen in table 1, which is 1%, 3%, 5%, and 10%. And then after the HEMA BA mix is obtained, it undergoes a process called emulsifier-free in situ emulsion polymerization technique with the addition of KPS, potassium persulfate, as the initiator of the polymerization process. And then after the polymerization process, the resulting composite hydrogel is washed with distilled water to eliminate free monomers, cut into this to obtain the contact lens shapes, which is circular shapes, and then it is dried at room temperature. Next. This is an illustration of the process from the HEMA solution and mixed with boric acid particles and then added with initiator polymerized to form the composite hydrogel. And next.
So after fabricating the contact lens, the researchers are assessing the properties of the material that they made. So first is the morphology and water contact angle. As we have known earlier, after Anjali's presentation, the boric acid is the independent variable, so it is the one that they change. And here are the parameters. Here, this is the morphology when using only 1% of boric acid, and this is the morphology of the hydrogel when using 10% of the boric acid. As we can see, it has a rougher surface in the 10% boric acid. So it means that increase in boric acid means that the morphology is rougher. And then next is the water contact angle. So a water contact angle is associated with wettability. So this is how we test water contact angle. If the angle is greater than 90 degree, then it has negligible, sorry, bentar, ini presentasi. It has, negligible wetting. When it is equals to 90 degree, it is also not, it doesn't has a good wettability, but if it is lower than 90 degree, it means that the material here could absorb the water and the angle become lower than 90 degree. So as we can see here, this is the results. The increasing boric acid means that show that the contact lens has lower contact angle, water contact angle. This is important. Why? Because in contact lens, it needs to have a good wettability, because if it is dry, yeah, if you have ever used contact lens, you have to apply eye drop once every several hours. And yeah, that is why it should be kept wet. And when it is wet, oxygen could also be transmitted into the eyes as also the nutrients. Next is the degradation. From the graph here, we could see that the higher the boric acid, this one, the orange one, the faster it degrades. And then on the right side is the swelling graph. This is the comparison between the FEMA with the boric acid and not with the boric acid. It only has a slight increase in the swelling. Swelling is, once again, it is important. Why? Because water needs to be kayak ditampung di dalam inside that hydrogel, so that oxygen could pass through the eye. Next is the oxygen permeability and the transparency. Yeah, these are both important parameters. Yeah. As for the oxygen permeability, I have explained earlier. And as for the transparency, our contact lens should be transparent, so we could see what. But if the materials are blocking our vision, then the contact lens couldn't be used. And this is the oxygen permeability. This is interesting. Why? Because as we can see, with increased boric acid, the permeability also increased, but then it decreased after 5 and 10%. Why? The researchers said that because the increased cross-linking density inside the hydrogel increased the dimensional stability, and there's also an increase in dimensional stability. It decreased the oxygen permeability because it is already packed up inside the hydrogel. As for the transparency, the higher the concentration of boric acid, the higher the concentration of boric acid, the lower it can transmit light. Next is the self-viability and antibacterial activity. This is important. Why? Because this is is it biocompatible or not to our eyes? And for the antibacterial, it is also an important parameter, because there shouldn't be any bacteria growing in or surrounding our eyes, because it could cause infection. This is the self-viability, which is tested by this diffusion method with different types of bacteria. As we can see, I think it's not that clear, but we can see the circle here. It is getting bigger, the diameter, because at the middle here is the hydrogel, and if the diameter is getting bigger, it it is caused by the boric acid of the hydrogel that could kill this bacteria. And this is the cell viability. It was tested using MDT assay, and it could be see here that there is no difference in the cell viability when of the FEMA BA, but there is a decrease of cell viability in the 5% and 10%. This means that only the 1 and 3% is eligible to be used at in vivo. Anjali, ini. Okay.
So in conclusion, the fabrication process used to produce the contact lens is called polymerization, which is initiated by the addition of potassium persulfate. And the FIMA boric acid composite hydrogel is possible to be used as a soft contact lens material, and that the ideal boric acid value is the 3% boric acid.
That's all from us. Thank you.
Thank you, Tanita and Anjali. I think very clear presentation. Group 1, please ask question, Ezra and Andy.
So what is the effect of water contact angle to the eye?
Sorry, could you repeat the?
What is the effect of water contact angle to the eye?
Water contact angle itu it indicates the wettability. Wettability itu that's like kayak betapa dia bisa, gimana dia bisa kayak maintain the water inside contact lens. Ya, if you have ever wear contact lens, kan kalau kering gak enak. And because the oxygen couldn't pass and couldn't be transmitted into our eyes. So that's why it is measured here using water contact angle. Now this is how they measure using the water contact angle. If it is greater than 90 degrees, this angle here, that means that the water couldn't be soluble to this material. Yeah.
Okay.
Any other questions, Andy?
Could you please go to the second last?
Nih.
Yeah. Why when the FEMA BA percentage increased, the cell viability decreased?
I think that's because of apa tuh? Wait, I'll remember the. I think it's because of the polymer here, the FEMA, because as we could see, the BA here doesn't, there is no causes caused by the BA. So I think it's the FEMA. Tunggu.
The FIMA kan kontennya sama boric acid, so the boric acid.
Yeah, the boric acid is the one that caused the cell viability to decrease. I think because too much of the boric acid could disrupt the cell and thus making the viability lower, kayak ngebunuh selnya.
Okay.
Okay, my question is actually, so the novelty here is they put boric acids in order to have antibacterial activity. Is it the main purpose of their research?
Yes.
Okay, so what is the problem with FIMA only contact lens?
Anjali, you want to answer or I'll answer?
The the boric acid, aside from antibacterial activity, it is also a cross-linking agent, sir. So it improves the structure of the polymer.
Of the hydrogel. Yeah.
Yeah, the hydrogel.
But without boric acid, actually, it can still make the polymer can still be contact lens, right?
Yeah, but another advantage of using boric acid is the this one, the wettability, because it is an important parameter. This one
Okay.
is because with the BA.
With
Only FEMA.
if it were only with FEMA, then it is easily it becomes dry easily.
Mm-hmm. Because the BA is polar, and it is soluble with water, and it could also help in holding the water at the contact lens.
Okay. Final questions will be: can we make it in our lab?
Sorry, sir. What's
Can we make it in our lab?
Bisa nggak?
I think it's possible because it's not very the equipment is not very complicated. Only like stirrer, and the maximum temperature that is needed is like 80 degrees. So
Okay.
yeah,
If
I think it's possible.
we are going to reproduce this research, what other improvement that you can suggest to improve, so as a novelty, to improve its quality? If we were to repeat again this in our lab, we make this again, what kind of improvement that we can introduce to increase its property, mechanical by good mechanical properties or whatever? What can you suggest?
I think if it's from the wettability, I think it's already good enough. Maybe the degradation time here, because it's only
Because what, it degrades easily?
Not that easily, because it still has a 80% remaining weight at the 6th week, but I think that could be improved. And then what else? I think adding other features, actually it is not related to the material sih, kayak di yang contact lens yang kayak yang sophisticated one, yang bisa not only for vision but also other usage.
For example?
Itu yang I have read the paper, but I don't know how to explain. Kayak we could see itu for example, our phone screen or what, using the contact lens. Yang futuristic contact lens. But that's no connection with the materials.
So but I mean, what do you mean? So is there any electronics inside or what?
There should be an
embedded electronics, but is it easy to make or not?
No.
Very futuristic, sir.
Yeah. Okay. So thank you, group 2, Anita and Anjali. So for this later, make your report, and also in your report, try to give a suggestion whether whatever you are reviewing can be reproduced inside our lab at SGU. Okay. If yes, then what kind of improvement also that we can introduce as novelty to that particular research that can increase the quality, the properties, improve the properties, etc. Okay, so don't forget this point in your report, because I would like to see that also. Okay, thank you very much.